Good. So today we're gonna discuss microbial physiology. What we need to keep in mind whenever we are talking about any living organism, this is actually what defines the living organism as an organism. Presence of physiology. So whenever you have any doubts, is it a microbial? Is it an organism or not? Is it a living structure or not? You ask yourself, does it have any physiology? Because what also unites all the living organisms, they're made of organic matter, lipids, proteins, carbohydrates. What also unites all the living organisms, the biggest volume, the biggest mass of the entire organism all the time will be water. Also, all of them will have genetical material, but not. But some of them could not have any physiology, could not have any metabolism. As a result, we cannot call them living organisms. And we'll discuss about them on other classes. But today we're gonna talk specifically about physiology and metabolism of bacteria. What includes physiology and metabolism of bacteria? First of all, in order to have a metabolism, you need to have organs, if we're talking about macroorganisms, and organelle, if we're talking about microorganisms, that are systems or structures that are responsible responsible on these metabolic processes. And in most of cases, these organs or organelles are similar in the entire living world, with small differences, right? So whenever we are talking about microscopical world, we start from dividing them into main domains, eukaryotes and prokaryotes. And here you can already describe some differences between them and what is in common between them. If we are talking about unicellular, it is much easier to compare unicellular eukaryotic cells and or organisms and unicellular prokaryotic cells. To be more specific, we actually don't need to specify that prokaryotes are unicellular, because all of prokaryotic organisms are unicellular. Meanwhile, eukaryotic cells could be both uni and multicellular organisms. So what is in common? They will have some external and some internal structures. External structures may play a role mostly of protection, shaping, and transportation. Meanwhile, internal, all the chemical, most of chemical processes, because if we compare their metabolism with ours, part of metabolic processes are extracellular, and part of them are intracellular. Because everything they have is their cell. If we compare microorganisms with macroorganisms, for example, bacteria with humans, what is in common? Both are living organisms, both have metabolism, both have genetical material, both are made of organic matter, but bacteria will not have all systems we have. Bacteria will not have all organs we have, because we are multicellular, we are multi-tissue, we have different tissues, different organs, different systems, extremely complicated organism. And all metabolic processes are happening only inside of the body. All the processing of like digestion and so on happens inside the body. Respiration happens inside of the body. Meanwhile, here the same thing, inside. But bacteria, in order to process something, in order to digest something, it eliminates some enzymes outside of the cell, around. It's like cooking for bacteria. It eliminates some enzymes. As a result, it processes some substances. After these substances are decomposed, it is able, by using different transport features, mechanisms, is able to absorb, to introduce all the nutrients inside. So digestion processes in bacteria are happening around bacteria, not inside, in comparison with macroorganisms.
Question.
Yeah.
Are mesosomes technically for extracellular digestion?
Mesosomes. Let's come back to mesosomes. Mesosomes. Seems we never escape them. Talking about mesosomes, they're used to be, they've been considered as organelle or parts of bacteria, but actually they're not. It is artificial structure.
It's just a change in the shape of the membrane? Or
Yeah. Like when mesosomes have been discovered, when electron microscopy started being used, and in order to make electron microscopy and in order to make most of types of microscopy, except for example dark field microscopy, where we don't make any fixation of the smear because we need to see the motility of bacteria. So as for light microscope, as for electron microscope, we need to make fixation of the studied material. So whenever we are making fixation of the material, for electron microscopy is applied chemical mechanism, chemical way. So mesosomes are actually deformations of the cell wall. Like because of these chemical substances, the bacteria contracts, and in some places, usually in place of attachment to the surface, it makes kind of invagination or something like that. And this cavity makes these mesosomes that actually are nothing but you can compare them with wrinkles made of chemical substance. So since this structure appears just as a result of fixation, it doesn't have any function. Because before this fixation, it never existed.
It says by the late 70s, they are no longer considered the part of normal structured bacterial cells.
Yeah.
So like 50 years ago.
Yeah.
It was
Okay, but do they still form mesosomes occasionally for digestion?
We can discover mesosomes, but they are not they are not a structure of bacteria, natural structure of bacteria. It is like, somebody died,
Okay, so
and something happened post-mortem. And you make a council. Which is the function of this structure that appeared after a human died? There is no any function. There is no any. During his life, he never used this part.
And now he's functionless. He's dead.
Yes, exactly.
Can't do anything.
So fixation is equal with death. Whenever we are talking about fixation in microbiology, is equal with death, because the processes that happen to the cell during fixation are incompatible with the life. If we are talking about fixation with physical factor, that is fire, it dies, it melts, it attached to the slide. That's it. If we are talking about chemicals, the same thing. It stops all the potentially present physiological processes in that cell. Okay. So, all the metabolic processes can be divided in two main groups: anabolic and catabolic processes. And I guess you've been discussing about that at school. You've been discussing about that on biophysics, physiology, biochemistry, exactly. And talking about catabolic processes, shortly, it's a process of discompounding of the material. And anabolic processes is a process of building of material. Now, all of these processes will be united by following elements. In case of catabolic processes, we have a molecule of something, of organic substance. It could be protein, it could be lipid, it could be carbohydrate. It depends on specific and on every specific process. As a result of some chemical reactions, the molecule of organic substance will be dissolved, discompounded, and as a result, we will have different smaller units and energy release. So it would be wrong to consider, because sometimes we are exposed to think that catabolic processes are leading to death and anabolic processes are leading to life. Actually not. Both of them are extremely important for life. Because catabolic processes, they actually have as target energy production, energy release. Meanwhile, anabolic processes are having as function as target building of some structures. So if we take a molecule, we dissolve it by using some chemical reactions, we have energy release and we have smaller particles. When we are talking about anabolic processes, we have small units, we have energy, and all together they are able to build new molecules. Bless you. Now, what also is characteristic for metabolic processes is transportation. For example, the bacteria, just as we mentioned previously, bacteria needs to consume something. As a result, bacteria produces a chemical substance, an enzyme, that is able to make some catabolic processes around bacteria. So bacteria releases enzymes around. It dissolves polypeptides and monopeptides, polysaccharides and monosaccharides, lipids in fatty acids, and so on. After that, by using different ways of transportation, like active and passive transport. Whenever we are talking about active transport, these are pumps. Why it is active? Because it works with energy. And if you have energy, it works. If you don't have energy, it doesn't work. We have passive transport that is divided in two main groups, is diffusion and facilitated diffusion, that are the second one looks like something between active and passive. Because there are kinds of porins that are able to conduct little by little some units. And diffusion, it's like penetrating through the membrane. And some of these processes are happening outside the cell. In this case, energy production is a side effect, is not a target. A target is dissolving of small units in order to consume them. And of course, it will happen with release of energy. After that, bacteria will absorb somehow by active and passive mechanisms, will absorb all the units it will need for life. And after that, it will start inside anabolic processes by using its own energy, by using its own materials and materials that came inside, and by using its own energy, which is the source of energy in bacteria and actually in any living cell.
Organic compounds?
Source of energy.
What? Carbohydrates?
Organic compounds.
ATP?
ATP is the classical source of energy. Whenever we have lack of ATP, any microorganism, any macroorganism starts catabolic processes, if you remember, that have as a target obtaining of ATP from other sources, from other matters. So normally, or most simple way of production of ATP is processing of carbohydrates. But if you don't have any, it is possible also by dissolving lipids and proteins. It will induce, let's say, more expenses for the organism, but still, in order to survive, first of all, we need energy. Because all the processes, or all the catabolic and anabolic processes are happening with presence of ATP. So ATP is this compound till ADP and phosphate, right? And usually here the process stops, but sometimes, specifically in case of anaerobic respiration, if we don't have enough ATP, sometimes ADP can be processed till AMP, but not too often. And already inside of the cell, it starts anabolic processes that have as a target DNA replication. That means we need to make double amount of genetical material, double amount of actually everything in order to multiply, in order to divide, because all the living organisms, they have as a target not only to live but also to multiply and to transmit all the genetical material to generations to come. All of these processes are impossible without following like metabolism is impossible also without also respiration and fermentation. Which is the difference between the respiration and fermentation?
Fermentation is happening without the presence of oxygen.
Without
oxygen. Not really.
Actually, even respiration sometimes happens without oxygen. Yes. So if we are talking about respiration, we can have aerobic, anaerobic, facultative anaerobes, capnophils, and so on. What means aerobic? Oxidation with oxygen. Anaerobic oxidation without oxygen, usually as alternative for oxygen will be either sulfates, either CO2. And when we are talking about CO2, this specific type of oxidation we call respiration. We call capnophilic. CO2 is capnophilic respiration. And both of them, the main difference between respiration and oxidation, between respiration and fermentation is that respiration is complete oxidation. And fermentation, what means complete? Do you remember the final product of respiration?
Yes.
CO2 and water.
Yes.
CO2 and water. It is the mostly final product of any metabolic process is complete oxidation. Whenever we are talking about fermentation, it is incomplete oxidation. So the process of oxidation have place, but the final products will be CO2 and, for example, alcohol, CO2 and different acids like lactic acid. So it is incomplete oxidation. Whenever we are talking about anaerobic respiration in our body, which is non-physiological for us. So please don't misuse aerobic respiration of bacteria, anaerobic respiration of bacteria, and aerobic respiration of our cells and anaerobic respiration, because anaerobic for some bacteria is natural, anaerobic respiration for us is non-physiological, that leads to side effects. And side effects are incomplete oxidation, that is fermentation. This is the way how lactic acid is produced during anaerobic respiration in our body.
And Viscoacidosis. Metabolic acidosis.
Yes. So it is extremely important to keep in mind, as higher is physical activity, as higher should be oxygen supply. Because if the activity will increase but oxygen supply will not increase, respiration actually happens in every single cell in our body. In mitochondria, all these processes are happening, and if somehow the oxygen will not be enough, instead of oxidation, we'll have fermentation. We have this acidity. As a result, in small amounts, it produces unpleasant feelings. In bigger amounts, it can produce death of a lot of cells, tissues, and so on. And except these processes, we also have all of them are related to energy generation, oxidation, fermentation, and photosynthesis. And photosynthesis is conversion of light energy into chemical energy, and after that, already is used for internal needs. And we have actually multiplication. It is the final target of any organism. And whenever we are talking about bacteria, all of them are multiplying by binary fission. That means all the bacteria will need to consume some extra energy for all the catabolic and anabolic processes. It will start the entire process divides in following steps. First of all, there is parental cell that actually exists. The second step, inside the cell happens replication of genetical material, of DNA. After that, after we have two sets of genetical material, the cytoplasm starts dividing into separate parts, and the last step, the cell wall also starts making difference between different parts of cytoplasm. And after this will appear two daughter cells that start growing, because previously it used to be a big cell, and after that there are two daughter cells. They are small, and after that they start also consuming substances from around and multiplying in the same way. Here's a question for logic. Here's a question for logic. Let's imagine we have a tube with culture media. We have all the necessary conditions for bacteria to multiply. We have this binary fission. And we have one bacteria in the tube, just one. Two conditions we know. First one, the speed of binary fission of this bacteria is 1 cell per minute, one fission per minute. The second condition we know, in 1 hour, the tube will be full. For how long it will take to fill the tube till the middle?
15
This is the mostly boring thing about our students. It doesn't take for too long.
It doesn't take what?
Till the right answer. Yeah, that's right, 59 minutes. Because logic, not math. Okay. Now,
I want to just
yeah.
capnophilic respiration is when CO2 is the final electron acceptor. Carbon. Capnophilic respiration is.
Capnophilic respiration is when CO2 is preferred by bacteria for oxidation process. So some bacteria are able to multiply only in presence of CO2. Some bacteria are able to multiply only in presence of oxygen. Some only in absence of oxygen. And some bacteria they are facultative anaerobes. That means they actually prefer oxygen, but if there are no any oxygen, if there is anything else but oxygen, they are still able to multiply. Not that much, not that fast, but they are still able to multiply. This is what makes difference between strictly anaerobic bacteria, for which presence of oxygen is actually mortal, is fatal.
Bacillus anthracis, for example.
For example, Clostridia. All the Clostridia are anaerobes, and in order to kill them, that's enough only to let them.
But there's spores.
Yes, good. But here is something you should pay attention, because they are non-living structures. This is why they are able to survive in nature, because they are non-living structures. They're potentially living structures.
Like, I think so. If they're not living, that means they have no metabolism.
Right, exactly. They don't have any metabolism.
Not even base cells, very low, very low.
Nothing, absolutely nothing.
It's just inert.
Yeah.
Like a rock.
Yes. And you know why? We can compare spores with dehydrated fruits. And this is actually the way how spores are produced. There are two. This is the cell. It passes through this binary fission, and one cell starts eliminating all the water from inside, and another cell kind of swallows this second cell. So actually it dehydrates in very gentle conditions, and where is no water, we don't have any metabolic processes, we don't have any life. This is to what we pay attention, that the biggest mass of any living structure is water. All the time it will be, or in most of cases it will be over 80%. By some exceptions, but in most of cases it will be over 80%. So if you don't have enough water, it doesn't mean that that spores are completely dry, like 100%, but almost 100% dry. So the amount of water that is present inside of the spore is absolutely not enough for any kind of metabolic process. As a result, it is like dehydrated fruit. It is not useless. It has potential for rehydration and for continuing some metabolic processes. This is why dehydrated fruits are not spoiling.
Yeah.
You can put how much bacteria you want in dehydrated fruits, but they will not destroy and they will not spoil these bacteria because, these fruits, just because there is lack of water. As soon as you will put them some water, you will have fermentation processes and all the catabolic processes could ever happen to any any organic matter.
So let me get this straight. So after binary fission, one daughter cell or one daughter cell gets dehydrated, and the other cell just eats its daughter, its sister brother. It just eats it, and it becomes its spore.
Not eats. Yes.
Yeah, I know, but
Because to eat means to
metabolize. Yeah.
Yes.
Yeah, but engulf.
It engulfs and keeps it inside.
And then it becomes its spore.
Yeah.
Okay, they're basically the same structure because they're identical to each other.
Yeah. Okay. Well, actually, can say that's main part of introduction in microbial physiology, and the next part of it we will discuss next week. Okay.